それではこれより分科会1、人と動物の共通感
We'll
染
now begin Subcommittee 1, Zoonotic Infections,
このセッションの進行を務めていただく座長は
Coronavirus Disease
福岡
COVID-19,
大学病院
and the
腫瘍
chairs are
血液感染症内科准教授
Atsushi Dr. Atsushi Togawa, Associate Professor, Department of Oncology, Hematology, and Infectious Diseases, Fukuoka University Hospital, and Dr. Hiroshi Shimoda, Associate Professor, the Joint Faculty of Veterinary Medicine, Yamaguchi University. Dr. Togawa, Dr. Shimoda, the floor is yours.
それでは、人獣共通感染症
Now we would like to begin the subcommittee for zoonotic infections, coronavirus disease COVID-19.
Okay, now I'd like to welcome our first speaker, Dr. Vincent Munster, from National Institute of Allergy and Infectious Diseases, Rocky Mountain Laboratories. With the current COVID-19 pandemic, he is actively involved in the development of medical countermeasures and providing critical experiment data supporting direct public health decision and intervention. Okay, Dr. Munster, please start in your good time.
All right. Good morning, good evening, wherever you are. Here it's evening in Rocky Mountain Laboratories in the United States. Can everybody see my presentation? I take that as a yes. So today we'll be talking about the virus ecology from host reservoir to disease. And if we think about like where emergent infectious diseases come from, and we have been seeing quite a fair amount of these emergent infections over the last 10, 15 years, typically what we see a natural reservoir over here on this side. And today we're primarily focusing on bats. And for instance, this is the situation of Ebola virus, where we have a lot of migratory bats here, and the virus can spill over to great apes. But then we also have that spatial connectivity on one hand, more connected global world with large cities. We have the local rural population and village. And between a lot of these different dynamics, the spillover can occur from the wild reservoir into the human population. And that has to be due with a combination of host distribution, host density, prevalence, and intensity. And the more we change our environment, the higher the likelihood of these viruses which circulate in the natural reservoir get introduced in the human population. So if we look at the virus diversity in bats, and here we have kind of like an updated amount of all the virus families found in different bat species across the world, there's actually two viral families who really stand out. On one hand, the coronaviruses, of course, very, very well known currently. And then we have the paramyxovirus. And but paramyxovirus, of course, are the highly pathogenic the highly pathogenic bat virus, Nipah virus, and Hendra virus. So if we then look into a little bit more detail and we think about coronaviruses, and in the last 2 decades, obviously we had the first emergence of SARS-1 dating back in 2003 and 2004. The virus came from a natural reservoir, later on to be identified rhinolophus bats, got introduced into palm civets in wet marks, and then had a couple of introductions in the human population. These viruses typically have the propensity for nosocomial transmission, so very efficient transmission in hospital settings. And the outbreak was actually contained after roughly 10,000 cases with a 10% case fatality rate, so around 800-900 people succumbed to infection. Then in 2012, we had another introduction of a highly pathogenic coronavirus, in this case, the Middle East Respiratory Syndrome virus, syndrome coronavirus, in this case in Saudi Arabia, where it infected one fatal case in Riyadh. Then retrospectively, multiple cases were identified in Jordan. And after a lot of field work, the dromedary camel was actually identified as the natural reservoir for Middle East Respiratory Syndrome coronavirus. Again, very similar to SARS-1, a propensity for nosocomial transmission. Given that these viruses circulate in the dromedary camel population, there is still introduction of this particular virus into the human population. And then, of course, we had the introduction of SARS-CoV-2, where we still don't really know the real ancestor to this virus circulating in the bat population. And we still don't know whether there was like an intermediate reservoir, as was the case with, for instance, SARS-1 in Wuhan and in the wet markets. Again, nosocomial transmission, but of course the difference between SARS-1 and MERS and SARS-2 is that the high scale of human-to-human transmission, and obviously the pandemic. So if you then look at what kind of viruses circulating, what kind of host, we really see that these beta coronaviruses, alpha and beta coronaviruses, really have a propensity, and they have been detected globally in bat species on all the different continents besides Antarctica, because they don't have bats. And if we then look here, if we stratify that, this is an overview where we find the highest bat species diversity. And typically the highest bat species diversity we find on one hand in South America, particularly in Brazil over here, and then high density in Southeast Asia. If you then looked at sample species, so where everybody went to actually start sampling bat species to find these coronaviruses, we again see a very high density in where we actually find most bat species. And again, a lot of activity in Southeast Asia. Of course, this is very understandable given the propensity for SARS-1, which started in Southeast Asia, but also other bat viruses, like Nipah virus and Hendra virus in Australia. And then if we really find like where do we find these alpha and beta coronaviruses? Again, here we kind of see we don't have the highest diversity of bat species, but we do have the highest diversity of these alpha and beta coronaviruses here in Southeast Asia, really suggesting that this is a particular hotspot for these kind of viruses, although they have been identified also in Europe as well as like sub-Saharan Africa and South America. So I think like the emergence of SARS-1 really started this era of virus discovery, and particularly using next-gen sequencing, we have like a lot of different kind of signals of these coronaviruses. And in the last like 15 years, we've seen all these papers coming out of the identification of these alpha and beta coronavirus in the bat species. And typically not much is done after the sequencing. So even though we have all these genomic signatures of these viruses in bats, we actually don't really have a good idea of the cross-species zoonotic or even pandemic potential. So can it transmit to humans, other animals, and does it actually cause disease? So one of my postdocs in my lab, Dr. Michael Letko, and he now started his own lab in Washington State University, we started working together to really like start a pipeline in trying to identify what do these viruses actually do. So we mined the gen bank and really came down to like not that much, but from all these different sequences, we only found 29 different receptor binding domain sequences. So the receptor binding domain is the part of the spike glycoprotein which the virus used to attach to the receptors on the cell surface. So using that, and here we see a phylogenetic tree, and we typically see that the SARS-CoV-2 and SARS-CoV-2-like viruses are in clade 1. Then we have clade 2 and clade 3. We can actually use these sequences to construct pseudotype entry assays, and we can really show that these viruses in clade 1 use human ACE2, and suggesting that these ones typically have a quite high cross-species and zoonotic potential. So if we then took a little bit further, we actually did exactly the same when the first sequence of SARS-CoV-2 came around, and this work was done in under 10 days by Michael, and he was actually able to show that SARS-CoV-2 also used human ACE2 receptor very efficiently, even more efficiently than SARS-1. And using that, we can actually scale these kind of like sequence database and really start mining the sequences in GenBank to really start understanding which of these viruses have truly, to a certain degree, zoonotic potential. So which one do actually bind to human cell surface receptors and then can enter the human cells. And that's typically very clade specific. Those clade 1 viruses really seem to have that zoonotic potential, whereas clade 2 and clade 3 doesn't seem to have that potential. And then we can even like add additional layers to that assay by looking at proteases. So we know that, for instance, TMPRSS2 is very important in the entry of SARS-CoV-2. So using that, we're now studying all these different coronaviruses, and we can then take it further and really focusing at this part of the equation and trying to understand like how many of these viruses we find in bats truly are a problem. And we did that previously with MERS-CoV as well, using functional studies where we have this co-crystal model with the MERS receptor binding domain bind to the human receptor. In this case, it's the DPP4. So with MERS, it's not ACE2. And we can actually calculate how well the MERS receptor binding domain binds to the DPP4 of other species. And we did that because at that time, the natural reservoir for MERS-CoV wasn't known. So here we can kind of show that common livestock species, very common livestock species in the Middle East, actually have like quite a high propensity of binding to the MERS receptor binding domain. And then we can use again that pseudotype entry data, really showing that, for instance, the camel really has like entry as efficient, for instance, as rhesus macaque and humans. And then we can follow that up, for instance, by doing field studies. Another study we did is actually looking at the reservoir. So understanding the reservoir better and how these viruses interact. So pathogen-host interaction is very important. In this case, we actually inoculated bat species with Middle East respiratory syndrome virus. We actually show that they become infected, but they don't become sick, and they have a very dampened innate response, really suggesting that these viruses and the host really have some kind of trade-off in able to like transmit further, but not making the host too sick. So as I said previously, the real reservoir of Middle East respiratory syndrome virus is the dromedary camel. And I think it's very important to actually do these kind of studies in the natural reservoir. So we started doing dromedary camel studies here with one of my collaborators, Dick Bowen at Colorado State University. And basically what we saw is after inoculation of these dromedary camels, a high degree of shedding of infectious virus for roughly about 7 days. And we see quite some nasal discharge in these dromedary camels, and really showing that MERS-CoV is really like a common cold virus in dromedary camels. And it actually reminded me quite a lot of SARS-CoV-2. And then we took this kind of like knowledge back into the field and start looking at dromedary camel populations here in Jordan in the Middle East. And we were actually able to show that these dromedary camels in the Middle East are infected indeed with MERS-CoV. And we actually see that the prevalence in juvenile animals is way higher than in adult animals. And it's something we typically see with these kind of viruses. So obviously then we want to know like how well do these viruses transmit, and that's again something we can take back into the lab, and we see that the transmission between dromedary camels is very efficient. So if you infect your dromedary camels on one end and then add naive uninfected dromedary camels, typically these infections goes within days. So why is that important? It gives you kind of like a framework to design, for instance, vaccines not focused at the human population, but at blocking transmission of Middle East respiratory syndrome coronavirus in dromedary camels. And that's something we've been working on. So dromedary camels are very interesting. So this is the distribution because they kind of overlap with Bactrian camels. So dromedary camels, 1 hump, Bactrian camels, 2 hump. Both are largely domesticated animals, although there are a couple of wild population in the Gobi Desert. So we were very interested in, are these Bactrian camels also susceptible for Middle East respiratory syndrome coronaviruses? And yes, they are as susceptible as dromedary camels. We followed that up and we did serosurveillance studies in Mongolia. And indeed, we see seroprevalence in bactrian camels in Mongolia, really trying to like expand the knowledge and trying to get that data across to suggest that we have like here the potential for Middle East respiratory syndrome cases as well, and not just in the Middle East or in North Africa. So I think like we're now entering really the era of functional metagenomics and really trying to use all that data on virus discovery, for instance, in bats, but also in like numerous other species, but then trying to leverage that kind of data in functional screenings, and then potentially based on these functional screenings, trying to in vivo testing and develop interventions. And I think for that, what we need is really like a very comprehensive system of surveillance, pathogen discovery, and ecological field research. So these are some of the studies we're doing in Congo with hammer-headed fruit bats, where we're not only looking on one hand what kind of viruses they have, and typically these animals have Ebola virus, but as you can see here, we put like satellite telemetry collars on these bats to really understand their movement patterns as well and how they actually interact with the local human population. And then I think we also need to know more about the molecular determinants of zoonotic and cross-species transmission. So obviously, it's very easy to really dump all these sequences in GenBank, but what we need to know is like, do these viruses actually have signatures of potential zoonotic and cross-species potential? That's something we're currently developing, really kind of like high throughput assays. And then modeling emerging viruses in our natural reservoirs. We need to take that data back to the lab. This is a One Health conference, and obviously we need to know more about the natural reservoir, intermediate host, human host, conservation, and really trying to understand not only what is circulating in these populations, but how we can prevent future outbreaks. And then I think like one of the things we're really working on is like modeling this zoonotic and cross-species transmission to really leverage that kind of knowledge, because if you know how these viruses are transmitted or the risk of transmitted, it's actually more easy to develop suitable countermeasures. And then lastly, going to these countermeasures is something we're working on with SARS-CoV-2, where we have a true aerosol transmission model in our hamsters. And here we actually have UVC irradiation, really showing that we're using UVC, you can actually completely block transmission between SARS-CoV-2 infected hamsters, really suggesting that these kind of tools not only focus at SARS-CoV-2, but also at influenza and TB, could potentially really be good preemptive strategies and would be implementable in a large variety of different circumstances. And then lastly, this is my group in Montana. A lot of people to thank, in particular Michael, who did all the work on SARS-CoV-2, and a lot of different collaborators. And with that, I'm ready to take some questions.
Thank you, Dr. Master. So I have a couple of questions. So first one is that those viruses that can use human ACE2, probably you think that those can be the candidate for the next pandemic, right?
Yes. But it's a little bit more complicated. So previously, Shengli Shi in Wuhan identified, for instance, SCO14 and WFV1. These viruses also use human ACE2 as a receptor. Still, they don't actually cause a lot of disease compared to, for instance, SARS-CoV-1 and SARS-CoV-2. So we think like the receptor interaction is the first step. Then, of course, that virus still needs to be able to efficiently use the internal machinery in those cells, and particularly like the ability to block or interact with the human innate system. That's what we think is the second step. So what we're now thinking is like, on one hand, we need to categorize and catalog the receptors these viruses use, and then trying to understand, well, the receptor uses is the first step. What is the second step which actually allows them to replicate efficiently in humans or not? So I think yes, to the degree, yes, these viruses would be high risk, but it's only part of the story.
Okay, thank you very much. So how can we prevent, how do you think, can we prevent this kind of next pandemic?
Well, I think that's a very good question, and that's of course something we've been thinking about for a long time. And the next speaker, for instance, as well, because he's working on avian influenza viruses. In the end, I think a very good surveillance system in the human population where you would do syndromic surveillance so that you pick up every spillover event. That's on one end. So from like you need to be at the source when that happens, and you need to have the way to rapidly identify that novel virus. And that's of course, on one hand, is in certain areas relatively straightforward. For instance, in the Western world or in the US or in Japan. In other places, it actually might be quite cumbersome, right? Like if you look at like sub-Saharan Africa, most of those spillover events will go unnoticed. And typically people do get sick for a whole range of diseases. So I think it's not that easy, and we need to really think about very carefully. And obviously that's where the One Health concept comes in, because that holds true for domestic animal species, but also like wild animal species. We need to have like better syndromic surveillance, and not only, for instance, like in Western Europe or Japan or China, but also sub-Saharan Africa and other places. In the end, I think what's most important is if we find something, we need to react fast. And that's something which we've done previously, for instance, with Ebola virus, where it's really clear the time it takes between identifying the first case, the longer that takes, the bigger the outbreak. And I think that's the same with, for instance, SARS-CoV-2. That one was actually identified relatively fast. However, because that virus spread in the human population without not everybody getting sick, or even had like asymptomatic transmission before they get sick, it does show that our window to react is actually really, really narrow. And that's something we really need to think about. Like if we have something like SARS-CoV-2, what would we do better this time than last time? Because obviously we didn't do very well, but other countries obviously did.
Right. Okay, thank you very much. So take the countermeasure as soon as possible is the most important thing, I think.
Yeah, correct.
Okay, thank you very much. So we have a couple time. So one more question about how about the intermediate host? But we find many virus from bats, but there's always some intermediate host of virus before spill over to human. So what do you think about how can we identify it, or how can we find them?
Yeah, I think that's a very good question, because that actually makes it more trackable. And obviously it's not always the case. So for instance, with Nipah virus, we have two situations. On one hand, we had the outbreak in Malaysia, where it first went into pigs and then into humans. And then we have in Bangladesh, where it actually gets transmitted directly from the bats. But I think that is a big commonality when you look, for instance, at avian influenza viruses, and you look, for instance, at SARS-1, that often introduction in a livestock species really allows that virus to build up a certain kind of level that then transmission to human population is bigger. And I think that's actually quite trackable, because like if you identify basically your animal production in a country, then it should be relatively straightforward to do surveillance. And I think like what I said in the beginning, now with all the next gen sequencing tools, cost coming down, and a lot of surveillance activity, we're going to find way more viruses than we actually anticipated, also in our domestic animal population. The problem is then, how do you then weed out the ones who are going to be problematic from the ones which are actually don't have that like very high potential, for instance, causing disease either in livestock or in humans. And I think that's now one of the big questions. And of course, that's the same for avian influenza, for instance, as well.
Okay, thank you very much. Now it's time to move on to the next speaker. Thank you very much, Dr. Master.
You're welcome.
You. Okay, next speaker is Dr. Randy Albrecht from Department of Microbiology, Global Health, and Emerging Pathogens Institute, Icahn School of Medicine at Mount Sinai. Okay, Dr. Albrecht, please start in your good time.
Thank you very much. Thank you to the conference organizers and the Secretariat for the opportunity to present today. I will cover the One Health challenges of influenza viruses and coronaviruses with some emphasis on vaccines. First, I want to point out that the concept of One Health actually originated in New York City, which is where I currently work. And it derived from the Manhattan Principles, which stated there was a broader understanding of health and disease demands a unity of approach achievable only through a consilience of human, domestic animal, and wildlife health. And that's illustrated in the left of the slide. And today I'm talking about two pathogens in the One Health perspective. One is coronaviruses, which Dr. Munster just presented. And the other one is influenza viruses. And the concept of One Health is illustrated in this picture, where you can see it is an environmental scene of a farm. We have the human element, and then we have the avian element, the poultry. Whereas the health of the environment, such as air pollution, could impact the health of both humans and the poultry. Poultry also could impact the human health through zoonotic transmission of something like avian influenza. And so this is what we'll talk about today. So the One Health approach to influenza was emphasized in 2009, and really it can be applied to pathogens with pandemic potential. And there are 4 elements to a One Health approach to pathogens with pandemic potential. And again, this is something that Dr. Munster talked about in his presentation. Epidemiology and surveillance, transmission dynamics, understanding the molecular biology and the pathogenesis of the disease, and immunobiology and vaccine development. But there are some challenges that were stated in 2009 during this symposium that still hold true today. And then one is that the integration of One Health across the fields of animal and human health presents challenges. There's a major emphasis on human health, with animal health and sometimes being secondary. And it really requires a multi-agency collaboration, because we're talking about pathogens that really do travel across the globe quickly, and so requires a coordinated response at the federal or government level. And again, One Health approach to pandemics essentially has 3 elements: surveillance, outbreak, and then response. And this is really important because the World Organization for Animal Health has indicated that 60% of pathogens that cause human disease originate from domestic animals or wildlife, which actually sometimes will present challenges in terms of developing vaccines or preparedness for pandemics. And part of this recognition is that WHO has described disease X, in recognition that an outbreak may occur by an unknown pathogen. So we may be prepared best as we can, but there is some uncertainty as to what the next pandemic could be. And again, this really means that there is a need for government funding of programs, because this is something that requires a coordinated response, and that there is a strong need for pathogen surveillance. And really, again, these need to have country-specific or global preparedness plans to have these coordinated efforts. And this will not be something local. As we saw with the COVID-19 pandemic, it can go global really rapidly. So we do need a global approach to detect and limit disease emergence. So first, I'll focus a little bit on SARS coronaviruses or coronaviruses in general. But again, I will talk a little about influenza viruses. So on this slide, there are 3 different types of pandemic potentials: influenza viruses, paramyxoviruses, and coronaviruses. Influenza viruses are highlighted in blue, or do have an animal origin, so they do cause zoonotic events in which we see animal influenza viruses crossing over into humans. And this has resulted in pandemics. But we also see this with coronaviruses that again circulate in many animal species. And this has resulted in pandemics and zoonotic events. Fortunately, some of these, such as SARS-CoV and MERS-CoV, have not had a pronounced transmission as we have seen with SARS-CoV-2. But we do know in history that coronaviruses have resulted in pandemics that have transitioned into seasonal coronaviruses. So if you think about bats as a central reservoir, this slide illustrates the cross-species transmission of coronaviruses. Then again, viruses from bats can emerge and transmit through different animal species. And there is a broad genetic diversity in these different species. So on the right-hand side is a phylogenetic tree of the sarbecoviruses. So these are SARS-CoV-like or lineage of viruses. And it just illustrates the diversity of these viruses. And so if one starts to think about a broad or pan-coronavirus vaccine, you can see that this animal reservoir, this genetic diversity, will present problems and challenges for the development of a vaccine. Now again, if you look at the biogeographical distribution of bats, families, and coronaviruses, this is just illustrating by color coding that we can detect bats species throughout the world. They're on different continents and different regions across the world. And that if you look in actually what is recovered from coronaviruses from bats, you see that in the number of bat species that were sampled, that we can see that quite a few had bat coronaviruses that were isolated. So again, bats are a significant reservoir. They have a broad geographical distribution and really do present challenges in terms of thinking about potential vaccines. In addition to this, coronaviruses also have seasonality. We do have seasonal coronaviruses. We have known that the coronavirus from 2020 did have a impact on the seasonality or the distribution of the viruses, but they still persist. So even now, we see that seasonal coronaviruses are rebounding. So not only are we dealing with a pandemic of a coronavirus, we have other respiratory viruses, including seasonal coronaviruses, that are circulating. And so currently, there's a lot of effort to develop vaccines against COVID-19. And this is really targeting the spike protein, but there are different platforms that are being utilized to develop these vaccines. And now the idea is, how do we take this technology, how do we take this understanding and make vaccines that can address this diversity of coronaviruses that exists in the animal reservoir? And so that leads up to the idea of development of a universal coronavirus vaccine, with the ideal properties being one, that it prevents clinical disease. It prevents infection by all SARS-CoV lineage or MERS-CoV lineage viruses. It will prevent infection by viral drift or recombinant variants, and something that is immunogenic, even if you have pre-existing immunity. One of the limitations that we see from live attenuated influenza viruses is that pre-existing immunity will diminish replication of these live attenuated influenza viruses, so they're not as immunogenic. And the same could exist if we tried to develop a live attenuated vaccine for coronaviruses. We do not want them to be poorly immunogenic because of prior immunity diminishing their replication. But obviously, we do not want to have antibody-dependent enhancement of disease because of the vaccination inducing an immune response that actually exacerbates disease. So the United States government, National Institutes of Health, has funding programs out for development of these broadly or pan-coronavirus vaccine initiatives, with currently 3 projects that have been funded through this. Next, I wanted to talk a little bit more about influenza viruses. I like this slide because I think, as you can see on the left, at one point, everybody has felt like this. We have all had an experience of influenza, which results from infection of an influenza A virus that is schematically illustrated on the right-hand slide that has 8 genomic segments with inside an enveloped virus. And this is negative sense genomic elements, but as you can see, there are 8 distinct genomic elements. And we have in the world the distribution of different influenza A viruses as well as influenza B viruses that co-circulate. And so this is illustrated here where the influenza B viruses are in orange or brown and the influenza A viruses are in teal or blue. And it just illustrates the co-circulation of these viruses. And again, makes one consider how to approach a universal influenza vaccine. One of the challenges is that we have antigenic shift and antigenic drift. Antigenic shift is when an avian influenza virus and a seasonal human influenza virus infect the same cell, and these segmented genomes are allowed to recombine, and that results in a new pandemic virus with this rearranged genome. As the human population develops immunity to this new virus, that puts immune pressure on the virus to evolve. And this process of evolution we call antigenic drift. And so the gene constellation stays the same, but we see phenotypic differences that diminish the efficacy of immune responses. So this is one of the issues that we have to deal with when thinking about animal reservoirs of influenza, is a potential emergence of pandemic influenza. These reservoirs, again, the wildlife birds are the main reservoirs of avian influenza viruses, but we do see zoonotic events that occur with either transient infection and replication of avian influenza viruses, or sometimes we do have the emergence of pandemics. And so again, this presents a challenge in terms of a One Health perspective of influenza, is how do we control this diversity of influenza viruses in an avian population or wild birds. And so one of the consequences, again, that I've already illustrated is the emergence of pandemic influenza viruses. In recent human history, there have been 4 pandemics of influenza. The 1918 Spanish influenza occurred when an avian influenza virus jumped into humans. That resulted in a new virus in the human population that resulted in, again, a pandemic. In 1957, we had the Asian influenza pandemic, in which there was a co-infection of an avian influenza virus with a human influenza virus from this previous pandemic that allowed for reassortment of the viral genomes, resulting in a new influenza pandemic. In 1968, the process occurred again, where we had co-infection of an avian virus with a human virus, resulting in a new pandemic virus. And at the time of 2005, it was predicted that there would be a new pandemic, and we saw that in 2009 with the emergence of the pandemic H1N1 or swine origin H1N1. I like having the question mark here because it illustrates that we do not yet know what will be the next pandemic, but we can be sure there will be another pandemic of influenza. So it brings up the idea again of a universal influenza vaccine. This is a little bit more simply stated than for a pan-coronavirus vaccine, but it should confer protection against antigenically matched or drifted viruses. Hopefully, we would not have to have the annual influenza vaccination for this, and would mitigate or reduce the disease burden from a pandemic of influenza. Now, I will disclaim or confirm that there are other groups that are developing universal influenza vaccines, but I want to quickly discuss what Mount Sinai is doing in terms of development of a universal influenza vaccine. And for this, we are targeting the hemagglutinin protein. This is the main immunogenic protein in terms of vaccine efficacy or vaccine protection encoded by the virus. And our idea is that we can change out the globular head of the hemagglutinin and exchange it for another influenza hemagglutinin. And we do this 3 times. So this will be sequential immunization. And so on the first immunization, we have a unique globular head with a conserved stalk domain of the hemagglutinin. And so we have a little bit of antibody being produced from both regions. In our next immunization, we change the globular head. The color coordinated here is yellow, but we keep the same stalk domain. So we induce more antibodies to this stalk domain. And then we will change out again for the third immunization, the globular head. So we are limiting the antibody responses to the globular head, but we keep the same stalk domain so that we keep boosting or enhancing these antibody responses to the conserved stalk domain. And so this sequential immunization process is our approach to developing universal influenza vaccines. And to do this, we are using the ferret model. This is my favorite model for influenza. And we are doing a sequential immunization approach in the ferret model, again, with 3 different immunizations that have hemagglutinin that express different globular head domains, but the same conserved stalk domains. We are inducing antibody responses through the stalk domain. And for this, we're either using live attenuated influenza vaccine or an inactivated vaccine. And for the data that I'll quickly present, this is with an H1N1 influenza A virus challenge infection in the ferret model of influenza. And as we can see, this is viral titers from the nasal turbinates and viral titers from the olfactory bulb that, depending on our immunization regimen, that we can see a reduction in viral titers. So if you look at the nasal turbinates in animals that are naive, that were not immunized, we see high levels of viral replication. Whereas if we immunize with an approach that has both an LEIV immunization and an inactivated vaccine immunization, we see the greatest reduction in viral titers, suggesting that this is our best approach for inducing immunity through this sequential immunization regimen. And we see the same for olfactory bulb, and we also see the same for lung viral titers. So this approach is promising in inducing stalk reactive antibody responses and really the basis of our universal influenza vaccine. We have tested this with group 1 influenza A viruses, that is illustrated on the left-hand side of this phylogenetic tree. And now we are looking at doing the same for group 2 influenza A viruses. And so this will be our approach to a universal influenza vaccine targeting influenza A viruses. So just to bring it back to perspective, again, with a One Health approach to influenza, we have to keep in mind human and animals. By and large, there's a lot of research on developing vaccines for human influenza. The surveillance and vaccines and therapeutics for animals is not as robust as we see for humans. But there are some animal vaccines for animal influenza. We see this for canine influenza, swine influenza, and poultry. The problem is that if you look at swine influenza, Roughly, there's 677 million pigs globally. So how do you approach vaccination of this large population of pigs across the globe? And the same for poultry with 33 billion chickens globally. So there is a huge economic impact when we have outbreaks of animal influenza viruses, either in swine or in poultry. So there is currently a need for developing these vaccines, but there is a challenge in how you put this into practice given the size of the target population. One approach that we've also looked at is developing bivalent vaccines that might be able to provide protection against two pathogens, for example, Newcastle disease virus and influenza virus. But again, it will be challenges in how we address this large population of animals. And you compare that with human influenza vaccination coverage rates in 2020, the range estimates are from 6 to 86 of the population, greater than 65 were vaccinated. So even for humans, we do not have a high vaccination coverage rate consistently across the globe. And so if we have this challenge for humans, how effectively are we going to apply this to animal influenza virus vaccines? And so lastly, for One Health considerations for veterinary vaccines, I'd like to illustrate on the right-hand side again that for human vaccines, federal funding for research on clinical vaccines far outweighs federal funding for veterinary vaccines. There's a lot of surveillance, luckily, being done for animal influenza viruses, but in terms of vaccine development on the clinical side outweighs the veterinary vaccines. There are clear benefits of veterinary vaccines. If you think about the economic cost of having to eliminate populations of pigs or chickens that have been infected with influenza viruses. But there are some challenges. So veterinary and clinical vaccines should use different antigen design. And so there is a potential that a veterinary vaccine could induce immunity that drives evolution of an influenza virus that may prevent or weaken the effectiveness of human vaccines. So really, animal and human vaccines need to target different antigens so that we do not weaken the effectiveness of human vaccines. But there's clearly a benefit for veterinary vaccines in One Health perspective. So I'd like to conclude quickly by acknowledging those who have contributed to this work. And I also would like to acknowledge collaborators in Taiwan, Claire Liu and Wei Li Xu, who have also participated in some of these studies I have not talked about today, but Claire did help out a lot with the influenza vaccine studies in the ferret model. So with that, I would like to take any questions.
Thank you. Thank you very much, Dr. Alverhit. It seems a very wonderful approach to make a universal vaccine for influenza. So this approach can be applied to the other viruses like coronavirus or something, do you think?
Yeah, again, that's as I showed for the coronaviruses, the same concept applies. There will be a challenge with identifying what would be the best immunogenic target. And one approach is what is called reverse vaccinology, in which you vaccinate and then look at the antibody response. And the antibodies will help you define epitopes. And then you can look for what are the common epitopes, and then build a vaccine that targets those common epitopes. But the same thing for coronaviruses and the same thing for influenza viruses. There will be a challenge of that animal reservoir. So we will never eradicate these viruses. The best is that we can protect ourselves from the unknown emergence of pathogen X, being either coronavirus, influenza virus, Ebola virus, whatever in the future.
Okay, thank you very much. Now we're running out of time, so thank you very much, Dr.
Thank you.
Abraham. Thank you. それでは次の、演題に、移らせて
Let me move on to the next presentation. Next speaker would be given by Professor Makoto Kuroda, Director, Pathogen Genomics Center, National Institute of Infectious Diseases. It is a recorded presentation, SARS-CoV-2 Genome Surveillance in Japan.
よろしくお願いいたします。国立感染症研究所病原体ゲノム解析研究センターの黒田と申します。本日は新型コロナウイルスのゲノムサーベランスによる
I'm Kuroda from Pathogen Genomics Center, National Institute of Infectious Disease. I'd like to talk about the SARS-CoV-2 genome surveillance in Japan. Please note that the contents are based on personal views and do not represent the opinions of the organization to which I belong. I have no COI to disclose. In public health measures, I think it is fundamental and important to quickly identify patients through active epidemiological surveys or contact tracing. If genetic information on viruses and pathogens is available, molecular epidemiological analysis can be performed using such genome information. And the results of interviews in contact tracing can be further complemented and utilized. When genetic information or genome sequences are deciphered, what are they used for? For example, they are used in DNA analysis to determine the parent-child relationship or as evidence in criminal investigations. In case of SARS-CoV-2, they are used to trace infection links based on genome information matches or mismatches, to understand differences among clusters, to clearly distinguish differences between domestic and foreign countries, and in border control based on sequence differences. For individuals, genome sequence can be used to evaluate the risk of lifestyle diseases and to predict the future in advance. It can also be used to diagnose cancer and other intractable diseases, which may be based on comparison between healthy subjects and patients. With SARS-CoV-2, the characteristics and individuality of mutant strains can be evaluated by comparing their increased infectivity and evasion of immunity. How can we decipher the genomic information of SARS-CoV-2? This positive strand RNA virus has a very long sequence of 29.9 kilobase. It is amplified in 2 tubes by multiplex PCR to increase the sequence from one end to the other in an alternating manner. The amplified DNA fragments in these 2 tubes are decoded by next-generation sequences, and the sequences are linked to identify mutations by comparative analysis with the control strain, Wuhan HU1 in China. In order to make these genome information analyses possible, not only at the National Institute of Infectious Diseases, but also at multiple sites, we are holding technical training sessions for 84 regional institutes of health. For example, after summer 2020, after the first and second waves, we held the training sessions and the second and third sessions. The fourth session, because of the declaration of a state of emergency, we were not able to have people come over on site to the NIID from various locations. So we set up a system where they could learn through remote training, such as Zoom meeting.
After about 3 months of peace and quiet time, the Omicron variant is now rapidly spreading. But we recently completed the 8th session on January 12th. At present, 62 regional institutes of health as public laboratories can perform genome sequencing. Now, we are aiming to acquire complete genome information. As I will explain later, a complete sequence is preferable to explain the linkage or disconnection between patients or between clusters in epidemiological studies. When only partial sequences are available, a concern remains that the false accusations may occur. I think this is where Japan's sequencing technology comes in. We are proud of the fact that we deliver outcome of higher quality than that of other countries. In this way, genome sequences will be connected. As you can see here, the genome sequence is passed down from generation to generation like a family tree, from grandfather, grandmother to father, mother, children, grandchildren, and great-grandchildren. Such genetic relationships may be traced in genealogy in the form of inheritance of infection links. Mutations can be found in the genome about 1 base every 2 weeks. By connecting them together, we can start to see the distribution of the mutations. Here is an example of information obtained by piecing together a sequence in this way. This was 1 year and 11 months ago when a novel coronavirus outbreak in China had just entered Japan for the first time in Kanto region. It shows that there's one mutation in this one location, suggesting it came to Japan in about 2 weeks. After that, it spread through various festivals and other events in Hokkaido. We learned that one person in Kyushu had been infected. We received this sample and did sequencing. This person had participated in a festival in Hokkaido, which we were able to prove also from the genomic information. In other words, this is a case where the epidemiological survey at the public health center matched the genome information. Next, we learned at this point that the infection links spread radially from Wuhan in China, which is the center of the disease, to Hokuriku, Kansai, and Kanto. If you think about the links of infection, you will realize that the infections are not connected in one straight line. This is expressed in this network diagram of genome information. Another thing that can be said here is that there is not just one infection link in this Kanto region, but multiple links. So you may understand that it is not very meaningful to be rooted in a region. In the first big wave, which now we can say was very small, a number of people returning from Europe in March 2020 were the starting point for the increase. This is a network diagram up to June 2021. It is a connected diagram comparing the complete genome diagrams of 60,000 samples. For example, this is a network diagram of alpha variant. An identical coronavirus genome information would be aggregated into this one circle. The larger the circle, the larger the number of patients. In addition, the regions are color-coded from north to south. This shows that there are many people with different sequences in multiple regions, and that disease has developed to a national scale. If I may go back, there was the first wave from Wuhan, after which the second wave generated in summer of 2020. You can see a thin line here. There was an incubation period for a while, and then a big wave formed in this summer. Next, in winter of 2020, in the year end and the New Year holidays, there's a line coming from a different place again. In other words, the first, second, third waves are not connected, but rather different events are created from different starting points for each season and each wave. R1 strain with E484K, which was detected in many cases in Eastern Japan and is considered to be an inflow from overseas, can be seen as a line that came from another place and increased. Alpha variant also increased after spring of 2021. As you can see here, they spread radially. If one point cannot be controlled, it will spread further to cause damage.
被害へと拡大していくということが見て取れるかと思います。はい。で、国内
And now, this is Pango lineage. This represents molecular phylogeny of domestic and airport or quarantine samples by their identities. This chart shows the characteristics of waves 1, 2, 3, 4, and 5 by the end of September. Each major wave has its main lineage. In contrast, quarantine samples appear in various colors, meaning to say, multiple countries, multiple lineages were entering Japan, and they were captured clearly at quarantine. But alpha, which is in reddish brown, has been found in large numbers after February at quarantine. As a result, this became one big major wave. Next, March and onward, this magenta or bright pink circle appeared. This was one big wave. Then it dropped, and the second big wave appeared at quarantine. Just after middle of May, another big wave appeared at quarantine, overlapping with the second Delta wave. This was confirmed as Delta dominant fifth wave in Japan. In the network schema, the first cluster represented the first wave. You see a lot of small dots. The second wave appeared in metropolitan Tokyo area and then spread across the country. This is the magnified view, including quarantine Delta. The green indicates quarantine samples showing multiple lineages. Samples identified in Kansai, Kanto, Kyushu, and Chubu areas seems to say sporadic. They were not spreading. This meant containment in local areas worked. That's what this picture suggests. However, this one point of origin became the starting point, becoming major player of the massive Delta wave. It became the mainstream. If we leave an origin uncontained, it will cause the situation that we have seen so far. This Delta mainstream had two major centers, two different centers. On the left circle represents 2,171 positive samples. This center is linked to the next type. The red line indicates the link or relationship. That's what genome information suggests. There are 1,197 links, meaning they spread out to various places. You can see similar phenomenon on the right. Big ones created the bigger ones. That's the point. They spread nationwide That's what I can say. Looking at the left and right, especially right genomic clusters, big ones created the bigger ones, many bigger ones. We do not know what event triggered such a phenomena. We have not identified the trigger that we can say for sure, but the schema tells the spread leads to further spreads. We must do something not to make it bigger next time. That's the conclusion. Now, about Omicron. The quarantine circles of Alpha, Delta, and so far, they looked somewhat large. But this Omicron, voluminous detection and rapid increase have been reported at quarantine. You can see that clearly after 3 months of quiet period, it is rapidly increasing. After the European first wave, second, third, fourth, fifth, and now Omicron. Looking at Omicron so far, not many origins are found as a starting point. We see about 4 origins. Individual origin shows spread within the region. Thanks to the local efforts, they are converging, settling down. But one region, one origin is causing nationwide spread. It is spreading right now. That is this lineage. Again, I say even one origin can cause massive spread. It is particularly true for this infection. Genomic information network, they are linked like you can see on the left. Let's take a color-coded map of different areas. Also, we can break this down to the following. Our analysis suggests the spread intensifies within the region initially, like this red circle. The yellow circle does not exist in isolation. Human traffic connects different areas, but in many cases, local spread stays within the region. Of course, local efforts are behind this phenomena, I understand. Containment efforts of individual origin are essential. We must do that. That's how I feel. Genomic information is difficult to understand. They are highly specialized information. In order to communicate with health centers, municipalities, and government agencies, plain language or vocabulary common to the profession should be used so that they can appreciate the information in their own terms. For example, field interviews for epidemiological survey, collect information about individuals, and build relationship map of a hospital or karaoke and or care facilities. We may be able to superimpose our genomic findings to visualize potential link or links and paint overall picture. This is just an example of such scheme that we are currently developing and make them available to incorporate expertise of multiple disciplines. I believe this is an effective approach to control infection. Finally, infection link mapping with sequence match or mismatch information will assist quarantine and many others defending our society. We can also biologically assess and inform safety level of new variants when they emerge. That is all. This is the photo of my lab staff. It's a small team, and Everyone worked hard. I thank them all. My acknowledgement from north to south, private labs, public institutions, and I thank NIID members too. Thank you very much. Thank you for your attention. Dr. Kuroda, thank you very much. Now we would like to move on to the next presentation.
From now on, we switch as the chair of the session, and we'd like to move on. Next speaker is Hiroyasu Ishikura. Dr. Ishikura is a professor at the Department of Emergency and Critical Care Medicine, Faculty of Medicine, Fukuoka University, and also he's a head of Emergency Center at the Fukuoka University Hospital. In 2020, from the beginning of outbreak of COVID-19 cases, he was actively engaged in the treatment of patients with severe disease. This is a rare experience he is going to share today. That is why we asked Dr. Ishikura to speak about this. His title of the talk is Treatment of Critically Ill Patients with COVID-19 Pneumonia ARDS in Fukuoka University Hospital, ECMO Center. Thank you very much for your kind introduction. I am Ishikura from Fukuoka University.
I'm participating in this forum today because people want to understand how the COVID-19, specifically critically ill patients, are being treated in Japan, is of some interest. So I believe that I was selected as a representative of the clinicians. There is no COI. As you may well know, after 2000, respiratory infectious diseases due to virus have been occurring, including SARS-CoV-2 2002 and H1N1 flu pandemic in 2009, and also MERS in 2012. Every 5 years or so, some kind of some type of viral respiratory infectious disease causes pandemic. And the history of human is the history of battle with infections. And then in 2019, in Wuhan in China, SARS-CoV-2 infection started, which became a global pandemic. And this is the data as of yesterday. Number of infected in the world is close to 500 million, and we have close to 6 million deaths so far. And in Japan, we experienced the 6th wave, and compared to the 4th and 5th waves, the current wave of COVID-19 is something that we have never really experienced in terms of a number of infected cases. So severe cases are being managed by us, which means that ECMO is used. Why have I started to focus on ECMO, and have we introduced this ECMO so actively? Well, this goes back to the pandemic of 2009, H1N1. At this time, well, there's an organization called ELSO, International Organization of ECMO, and you can see all the nations who are members of ELSO. And at the time of 2009, no Japanese facility was a member of ELSO. We didn't have ECMO center in Japan. And as a natural result, among the key countries in the world, in terms of survival of ECMO-treated patients for H1N1, we were the lowest. Compared to Sweden, where ECMO was actively used, the survival was 1/3 in Japan. So we decided something had to be done about this. I flew over Mount Fuji, and in January 2011, I flew to Stockholm, Sweden. The reason is, as I said before, both in Europe and also globally, the center of ECMO, the leader of ECMO, was Karolinska University at the time. And I really wanted to see how things were being done and learn from them. And I was still a bit younger then. The gentleman in the middle with white shirt is Professor Palmer, who is really well-known authority. And then at the ECMO Center at Karolinska, this was the chest X-ray and also CT, H1N1 influenza cases. These are very similar to the X-ray and the CT of the COVID-19 patients that we're treating right now. So this was a severe case treated with ECMO. And I was really surprised to find out two important keywords, first of which was awake ECMO. Patients are staying awake with ECMO. So as you can see, this patient is on ECMO, but he's eating a candy bar. And we need to decrease the sedation. Comfort is important. And within that, maximize the comfort of the patient. And ECMO is done over long term, which means that respiratory management, well, oxygenation in the body is basically done by ECMO, and therefore the respiratory management is not that important. I don't think Japan had this type of strategy at the time. And this is hangar, and you can see the ECMO car, yellow, and to the right you can see a jet plane. Karolinska ECMO Center are managing these. So if the condition improves while the patient is on ECMO, what they can is transfer the patient back to the UK where the patient came from. So Dr. Parma himself is involved in transporting the patient into the jet plane with ECMO attached. And as you can see, he has a big smile on his face and he says, "I'm going to the UK now." And I asked how long it takes, and he said about 4 hours. Anyway, this kind of situation already existed in Sweden more than 10 years ago. So this is a second keyword that I learned, ECMO transport. In 2011, I visited the ECMO Center at Karolinska for the first time to study. And after that, in 2012, 2015, and 2017, we sent our staff there as well for them to be trained. And this was a small program, but I was convinced that we would have another pandemic of severe respiratory diseases caused by virus. And based on that understanding, we were continuing with this small program. And as expected, in February 2020, we had SARS-CoV-2 virus infection causing pneumonia. And this was a pandemic of pneumonia. Now, with that situation at the Fukuoka University Hospital, we decided to open our own ECMO center. And this is what it looks like. First in Kyushu. And in 2011, at the time, as you can see, highlighted in red circle, in Japan, we didn't have any ECMO center. But in 2021, ECMO center locations, we could find 7 of them in Japan already opened. And in Kyushu, Fukuoka University Hospital was registered as one of the ECMO centers. Now, It is very difficult for us to show how the patients get treated for severe case of COVID-19. I have a video. First of all, it's very important to provide prone position therapy. So lay the patient on their stomach. This patient is already on ventilation and also ECMO. Why do we need to put this patient this way? Well, this is actually quite logical if you think about it. On the left, you can see the CT scan, and the ventral side bilaterally, this is wide, meaning that there is no space for air to go in. And the alveoli on the dorsal side is already collapsed. And in this case, if you try to ventilate the patient, the ventral alveoli would get overdistended and ventilation would be uneven, which also means blood flow would be uneven. Blood is liquid, and therefore it is pulled by gravity and tends to travel to the dorsal side of the patient's body. And the ventral side blood flow will be weakened. So in terms of ventilation and blood flow, we would see some disparity or imbalance. In order to solve this, very simply, we can just reverse the position of the patient and put the patient in a prone position so that the collapsed dorsal alveoli can be improved, and also overdistended ventral alveoli could be improved. And in prone position, the status can be improved, and this is why prone positioning is actively used. For severe respiratory failure patient, this is a very important method for us to use. Another thing that is important is in this prone position, together with the respirator movements, press the thoracic cavity in order to help with respiratory rehabilitation. So this is one of the important for respiratory failure in severe cases. So we put patients in a prone position. Sometimes it shows improvement in ventilation, but sometimes that is not enough. In such a case, we introduce ECMO. ECMO has a very simple structure. From the body, we have these tubes to remove blood from patients, and then we have a blood pump. This closed circuit, we have a membrane oxygenator, that is artificial lung, to give oxygen to the blood. So blood go through this circle outside the body, and in this process we introduce membrane to give oxygen to the blood. Within this membrane structure, we have oxygen, air, and carbon dioxide. And using this gas blender, we adjust the ratio so that the most appropriate oxygen or carbon dioxide can be provided to the patients. Carbon dioxide accumulated in patient's body are removed, and oxygen is to be delivered to the patient. This is a patient who's put on ECMO, and the two tubes are in the vein, and the oxygenated blood coming from ECMO are introduced to the patient's body. Now, during ECMO is used, as I said, ventilation is not so important.
As with severe pneumonia with COVID-19, in a severe phase, the inflammation in the pneumonia is so severe, and we don't want lung to work too much. So when we put patients on ECMO, as you can see, tidal volume is almost around 100. That is 1/5 of the usual amount. So oxygenation is pretty much dependent on ECMO. So that's another keyword, lung rest. When ECMO is used, patient lung should not be used as much as possible. During the use of ECMO, as I said earlier, we do the awake ECMO. We decrease the sedation and we make sure the patient is in a comfortable state. We let the patients to eat jelly, as shown here. This patient is very obese. The body weight is over 200 kilograms. It's not good to such obese patients in a lying position. So we do the wake ECMO and try to start rehabilitation as soon as possible. If we can do the ECMO successfully, such rehabilitation is also possible. I said patient's comfort is so important. Soon in Japan, we are going to have a spring cherry blossom season. This is March 29th last year. And we had a very nice cherry blossoms blooming in front of the hospital. So we let the patients outside of the ward to enjoy some blossoms. So even though patients may be in a severe conditions, but of course we do the ventilation control also. And if patient can go outside like this, that will help to patients to feel better. After about 1 month, the respiratory function is improved and the lung compliance is to be improved. Gradually, as you can see, the tidal volume can be increased. Now, I would like to show you epidemiology data of our ECMO cases up to December 31st last year. That is before we saw Omicron patients. The first, second, third waves of the outbreak, we had the 32 cases in total, and one of the patients used ECMO for 189 days, and he recovered so that he was able to go to the restroom by himself. And this was the first case we experienced with such a long duration of use of ECMO. And then after that, we had a fourth wave of a pandemic when we start to see the Delta variant. As you can see in hospital mortality column, probably due to the improved level of the care, or maybe thanks to the vaccination, we start to see the decrease of severe cases. And it seems that fewer patients were dying. And in the fifth wave of pandemic, here all the patients were due to the Delta variant. Unfortunately, we lost 2 patients here. Compared to up to third waves, if you see fourth wave and fifth wave, survival rate seems to be improved. So far we have had 69 ECMO cases, and now we have an Omicron outbreak from the beginning of this year, and we have 5 patients now on ECMO, and we see some severe cases. So in these 69 patients, severe COVID-19 pneumonia patients, among them 40 patients had ECMO. And the duration of the ECMO was 30.5 days on average, and the longest was 189 days. More than 80% of severe pneumonia patients were male. Average age is 58, and mostly they are in the 60s and 70s. PaO2 and FiO2 at the time of administration was less than 100, and it's 83 for ECMO cases. If you consider the usual amount is about 500, we can see the oxygenation is so poor in these cases. Survival rate of this total population is 78%, which is not bad, and 67.5% in ECMO cases. We would like to improve this survival rate even further. The left bars shows survival in each age group. The fatality is higher in older patients, as you may see in data in anywhere. You can see the average age of the patients in each wave. In the first wave and the fifth wave, we can see the average age is decreasing. I'm not sure if this is just due to the coincidence, but in my personal impression, because of vaccination was broadly done in elderly, particularly in a prioritized manner, and that may play a role. The far right is a fatality in each wave of a pandemic, and a recent fatality is lower compared to the first waves, for example. That is probably due to the benefits of vaccination, possibly. Another thing we have in our ECMO center is an ECMO car, which was introduced last year. ECMO car is a larger version of doctor car. Stretcher can be stored in the ECMO car with a couple personnel standing by. Oxygen and electric power and batteries can be put in as well, so it can cover a broad area transport. How did we introduce this ECMO car? Fukuoka is located in Kyushu, which is a large island in Japan. And when we have severe cases in different prefectures, we need to transport these patients beyond prefectural borders. So that is why we decided to introduce ECMO cars. We had this agreement covering 9 prefectures, including prefectures in Kyushu and Yamaguchi. The governors signed this agreement, and now we can transport patients with severe disease to Fukuoka University, and we did actually transport patients from nearby prefectures. Unfortunately, in Kyushu, we have still only one ECMO center at our Fukuoka University. So if we can consolidate such severe cases to ECMO center, what kind of benefit can we have? If you see this survival rate at the ECMO center and the non-ECMO center with conventional management, survival rate is improved by about 15 points by consolidating the severe respiratory failure cases care at the specialized ECMO center. So we are going to continue this patient's care in a specialized center. Many centers in Japan do not have sufficient know-how with ECMO yet. So we go to different prefectures in Kyushu to provide ECMO education and training. We visited centers a lot last year, 2020, but in 2021, we were able to visit only one location because we are rather busy with patients' care. But we would like to continue to provide education training in those other centers. Now, when it comes to the survival rate of severe COVID-19 patients with ECMO, the survival rate in the world is 52%, and in Japan it's 64%, and in our ECMO center, it's slightly better than that of Japan, 67.5%. So I believe I can say that the know-how and expertise we have accumulated helped us. In the end, I'd like to emphasize that the ECMO cannot be run only by physicians. So it requires different professionals and personnel. And so multidisciplinary staff help us to provide better patient care. And still, we are in a trial and error stage, and we'd like to save as many patients as possible. Thank you for your attention. Thank you very much, Dr. Ishikura. We have a couple of questions. First of all, as you mentioned earlier, ECMO is a treatment for lung rest. As you show in patient CT, it should take quite a long time for lung to regain its function. In your cases, usually, how long does it take on average? Well, when ECMO is not introduced, it usually takes 2 weeks, but when ECMO is used, at least 1 month is required to see a lung function recovery. So when ECMO is used, that means the patient is in a so severe condition. So again, it's very important to avoid the situation that patient's condition gets very severe. In your patients, of course, even after ventilation, the extubation or ECMO may be needed. For example, clinically, do you see some differences with regard to patients' recovery, for example? Well, this may not answer your question, but we see Omicron severe cases in 5 patients. 4 of them didn't receive vaccines. 1 patient had the RA and taking steroid. So Omicron, in order to, well, some people say that in Omicron variant, patients do not get severe so much, but is this due to the nature of the strain, or is this due to the vaccination? I wonder which is the case.
打たれた方で重症化される
If the patient is vaccinated, will they develop a severe or critical disease? We don't know. But obesity and lifestyle-related diseases, which are risk factors, if the patient has those, even after vaccination, they may develop critical or severe disease. According to my view, unvaccinated patients may be more prone to severe cases, and that's my experience with the Omicron. Thank you. Therefore, vaccination is extremely important in order to prevent severe symptoms. If the patient is on some kind of immunosuppressant, even after vaccination, maybe the effect of the vaccine is quite weak. In order to protect those people, healthy people need to vaccinate themselves in order to protect the other people for whom vaccine is not that effective. So we need to think of ourselves as a group, and it's very important in order to curb the pandemic to make sure that as many people as possible get vaccinated, including young people. So thank you very much for your kind presentation.
Next presentation, please, by Dr. Professor Satoshi Kutsuna. Dr. Kutsuna is currently with the Osaka University Graduate School of Medicine and Department of Infection Control, and he's the chief of the infection control at the Osaka University Medical Hospital as well. And Dr. Kutsuna has been in the previous post, has been globally quite active in research as well as the clinical. Dr. Kutsuna, please. Thank you, doctors, for giving me such a various opportunity to present my ideas on Omicron as well as the COVID-19 infection control. Let me share my screen, please. Let me start my presentation. I moved to Osaka University since July last year. This is my CI slide. And first of all, current infection status in brief. As you can see, these are the human transmissible COVID viruses or coronaviruses, and representative ones. SARS, MERS are the two predecessors which were rampant in the past 20 years amongst humans. Many people died because of those infectious diseases. Fatality was much higher than the new COVID-19. And this COVID-19 or new coronavirus, 400 million people have been infected. The scale of the infection is 5 digits higher than the previous waves or the previous diseases. 400 million people got infected, and more than 1% of the people infected died. Social impact. This is the greatest in the past 100 years as a pandemic, I would say. This is a massive scale indeed. This is the global trend or case counts. And for the past 2 years, we have seen waves after waves, ups and downs. 2021 December till January 2022, rapid increase of the infection cases. And this is unprecedented for this particular disease. In 1 week, let's say 5 million globally, this is called a massive pandemic, but currently 20 million in a week globally. That is a new case reports. This is abnormal, so to speak, unusual. And the Omicron made this spread. This is the Japanese situation. In Japan, mid-August was the previous peak of the 5th wave. It settled in October. So for the past 3 years, October, November, December, very low case counts. And towards the end of December, Omicron community infection started to spread, and it's been exploding, so to speak. For the past 1 week, the increase in case count has been slowing down, so to speak. We may now see a peak in this wave. Omicron, why? It's easy to spread compared to the others. Let me have some explanation on this. This is the United Kingdom data. Omicron compared to other variants, the speed of rise in infection explosion, so to speak. The largest number experienced was with the Delta variant. In UK, in order to reach the figure of 500,000, it took 150 days. In case of Omicron, reaching 500,000, it took only 50 days, 1/3 of the Delta, so to speak. So infectious, indeed, infectivity is quite high. Why? So much case counts and the rapid spread. There are several reasons. Let me explain this. One, infected patient may produce other infected patients. That is the generation period or incubation period, 5 days in the previous COVID viruses. So 5 days later, 1 patient will become 2 patients, and in 10 days, 4 patients. But in case of Omicron, this is only a shorter 3 days, 3 days and 2, and 6 days, 4, and 8 days, 32. So 8 times more by day 10, so to speak. And the more time passes by, you get much larger numbers. Another characteristics is that the vaccine is somewhat effective to Omicron, but the vaccine administrators may get infected with this virus as well. This is a previous Delta on top. 2-time vaccine takers, harder to get the next infection. So even when someone is infected around you, it is less likely to have a chain of infection spread. And after Delta, 3 months of quiet period I mentioned, that was when 80% of the Japanese population got vaccinated. So a small number of infected was not likely to spread to the others. However, when we changed over to Omicron, those who got vaccinated, I don't say it's totally ineffective, but somewhat effective is the correct way to say it. So they may get infected although they get vaccinated. Because of that, Omicron is increasing its case counts rapidly. Next, Omicron is said to be let's say milder than the others or less likely to get severe, so to speak. Compared to the Delta, overseas report says the hospitalization risk is 1/3 or 0.3 times higher severity risk, or so to speak. It's less likely to get severe. However, we have so many vaccinated population. So vaccinated population also get the Omicron, and overall it leveled off that level of severity is less, so to speak. And for unvaccinated people, compared to Delta, the hospitalization risk is 0.76-fold, so to speak. Delta was characterized as the severe infection compared to the previous ones. Alpha or others got infected. Perhaps the Omicron's severity trend would be similar to that of Alpha, it has been said. And let me try to explain this in plain mode. So I hope it's understandable. This is wave 5, fifth wave. Let's say the 50% got the one vaccination, and during the course, it get the second vaccination. So the breakdown of the population was half and half, vaccinated and twice vaccinated or unvaccinated. Those who got vaccinated, they're likely to get the infection. So those who got infected during this period was just a part of the vaccinated plus most of the unvaccinated, so to speak. So these are the composition of the Delta infection cases. So the virulence of the virus was attenuated, so to speak, with the vaccine. However, for the Omicron situation, 80% of the population got the vaccination twice already, but they get infected as well. So despite of the vaccination, Omicron gets the infection, meaning to say unvaccinated 20% of them and vaccinated people's 80% similar date. Then vaccinated people made it likely that the overall population's severity was less than the previous ones. That's how it's being explained. It's not the pure Omicron infectivity and virulence. It is easier to get infected, but you cannot say plainly that you do not get more severe with Omicron. It's not known. Perhaps the vaccine factor has not been well incorporated in the theory. And for your information, Omicron characteristics, clinically, I would say clinically, throat and runny nose are the major symptoms. Many say that. Compared to the previous variants, it's closer to common cold or influenza, or the COVID-specific abnormal taste and the smell are much reduced. So symptoms cannot tell you whether you got COVID or common flu, common cold or flu.
Next, I would like to talk about how to control the transmission. In COVID-19, there are 3 routes. One is contact. By touching something that is covered with virus or somebody's hand, and then touching your own eyes or nose, mouth. Before you touch your face, you should wash your hands. This is very important. Another thing that is another route is droplets. People who are coughing or sneezing spread the infection with influenza, but asymptomatic people with COVID-19 also have a lot of virus. And the virus is contained in large quantity in saliva. So any droplet, including saliva, also contains a lot of virus. And this is why the transmission occurs quite easily between people who talk with each other without wearing masks. Another route is aerosol, smaller than droplets. Especially in a closed, unventilated space, beyond the distance of droplets of 1 to 2 meters, transmission may take place. So contact, droplets, and aerosol are the 3 important routes of infection or transmission. And as I mentioned before, peak of infectivity exists before and after the onset of the symptoms. And in the case of Omicron, maybe this is shifted slightly to post-onset of symptoms. But still, before the onset of symptoms, you may be spreading the disease, which makes this infection quite unique. So if people don't have symptoms, how do they spread the virus? Please watch this video.
Now I'm recording. Stay healthy. Great. Stay healthy. Great. Less loud. Stay healthy. Good.
このようにですね、普通にその会話をするだけでもですね、
As you can see, even with a normal conversation, many droplets fly into the air. Mask can cover the droplets during conversation. This is an important countermeasure. If you sing loudly, for example, if you do karaoke or chorus, I know that we have had clusters in those situations. Transmission is very likely to take place. And masks can reduce the amount of droplets by half. So with or without symptoms, it's important to wear masks.
もう一つはですね、3密対策ということで、
Another thing that is important is making sure that the 3 C's are avoided. This was early a COVID pandemic period, and the people praying in a hall. As you can see, it is probably not ventilated. This is a crowded place, and they're praying together without wearing masks, producing a lot of droplets. This was a cluster of more than 5,000 people in this particular location. The 3 C's as a concept, I believe, has already taken root. But in the early days, we were saying that the overlap of these 3 C's is the most dangerous. But now we have Omicron with a strong, high infectivity. So we need to avoid any of the 3 C's. And the other thing is hand washing. Frequent hand washing is important to control the spread. And I'm sure that you have been doing this already as a part of general practice, and I hope that this practice will continue at a high frequency. Last but not least, significance of vaccination. Vaccination against Omicron. People say that vaccination doesn't work as well for Omicron. And compared to the other variants, it's true. Vaccine is weaker in preventing the onset. After 2 Pfizer shots, well, it used to be 90% protection, but now it's 60%, just above 60%, and it goes down over time. But with booster, again, the level of protection goes up. Compared to the vaccine naive, those who had 2 shots versus 3 shots, as you can see, the number of infections is lower in the last 2 groups. Vaccination and booster shots are important in preventing the spread, and also in terms of prevention of hospitalization. The gap is even bigger between vaccine naive versus those vaccinated. So vaccination is effective against Omicron, especially in prevention of hospitalization. So the protection for the effectiveness is being maintained. In terms of adverse reaction, this is data from the United States. Compared to the second shot, this is the second shot. The third shot, in terms of malaise, headache, and the fever, we see fewer incidents. And this is Pfizer, this is Moderna. And those for Moderna booster shot is half of the previous shot. And the adverse reaction is also weaker. There is excess inventory of Moderna. Effectiveness is higher with Moderna, and adverse reaction is not much different. So I hope that Moderna vaccine will be used to vaccinate, especially the elderly. We are now in the midst of the 6th wave, which is about to peak. Omicron is very infectious, but less likely to cause severe cases compared to Delta. Wear masks and avoid the 3 Cs. This is basically the same for Omicron as the other strains, and the vaccination is still effective in preventing severe illness. So I really hope that the people would consider the booster shot, especially in the elderly population. That's all from me. Thank you. Thank you very much for your very valuable presentation.
Dr.
Kutsuna, thank you very much. Because of the time constraint, I would like to have just one question. This has been going on for 2 years almost, and we have to consider the exit strategy. As you know, UK and European countries started to lift the mask rules. In Japan, with more advanced vaccination and the coverage rate, do you think we can use less mask or no mask so that we would go back to the previous lifestyle? Your personal comment is highly welcomed. Thank you for the question. Let's see, Denmark, for example, the elderly's booster shot has been given in more than 90%. So in advanced countries, in terms of vaccination coverage, they started the lifting of the mask rules, so to speak. So In Japan, prerequisite is the elderly's completion of the booster shots, because we see high fatalities in the elderlies, like 100 or 200s, and they are not getting the booster shots in time, so to speak. We should promote booster shots for the elderly first, and then discuss the exit strategy. And of course, the UK discussed and exercised the lift of mandatory masks, but they have to retract and go back to the mask rules and lift it again and so on. So you have to see the recurrence of the similar phenomena, and you have to accept a certain setback, so to speak. But that discussion should be done right after the good enough booster shot coverage. Thank you very much for your comment, and we appreciate your time and talk. We would like to hear from you again in the future. Thank you very much for your presentation today. Thank you. So this is the Subcommittee 1, Zoonotic infections. This concludes the morning program. Thank you for your attention.